Okay, so welcome everyone. So our speaker today, Dr. Kalinski, is a postdoc from Rhodes University, and he's just returned from Metabolomics in Canada, where he shared this data he's going to tell us all about. So a brief background or introduction of who this man is. So he does LC-MS based untargeted metabolomics. And he says it's a powerful tool for environmental monitoring of xenobiotics. So in the study, he applied this approach to analyze dissolved organic matter, which he calls DOM, from aquatic samples. And yeah, they're collected in Algoa Bay in South Africa and its tributaries, the Swartkops and the Sundays rivers, which represent urban and agriculture impacted estuaries, respectively. So the aim was to assess the utility of untargeted metabolomics to annotate xenobiotics of concern, as well as their distribution in DOM within the Algoa Bay system. So he employed reverse phase UHPLC-MS and positive mode electrospray ionization on a quadrupole-Orbitrap mass spec for untargeted screening of xenobiotics and approximate quantification with authentic standards of various compounds given there. I think he'll give us all the full names and correct pronunciations of them. And he's going to talk about his findings, how they showcase the efficacy of untargeted metabolomics approaches in environmental monitoring of aquatic systems. So it's always nice to see the diversity of metabolomics, especially now in environmental sciences in this presentation. So I'll hand it over to Dr. Kalinski. So please do share with us your findings of your study.
Thanks, Shane. Thanks for the introduction. So I'm going to talk today about a project that we started a couple of years ago on dissolved organic matter analysis from aquatic samples in Algoa Bay. And first, I'll give you like a little bit of background to our group, and then dive into the study at hand. So first of all, this is actually our group here on the left that you see. We're basically a group of chemists, microbiologists, geneticists. And we funded, our main grant at the moment is through the ADD Hub, so it's antimicrobial drug discovery. And we started off working on marine invertebrates a couple of years back, and then now we've also gone into marine microbes from marine invertebrates and other sources, and basically try to find new antibiotics from these organisms. And we started using tandem mass spectrometry, really just from the point of view to see which samples are different to each other. So we don't prioritize the same samples that produce the same type of compounds. And also to de-replicate, let's say we have some active samples and now we can detect known antibiotics in there, then they may be less interesting for us. Yeah, and we basically work with a host of collaborators, and the guys you see on the right here, Xavier Sibinundu, Gwyneth Matchett, Tommy Bornman, and Janine Adams, they're all based in South Africa. And they're involved on the Dissolved Organic Matter project, and then as well, very importantly, Daniel Petras and Peter Dorestein. Daniel Petras, he was a postdoc of Peter Dorestein at University of California, San Diego, and now he's a PI of his own in Tübingen, in Germany. So they basically kicked off the DOM project. All right, so here, this is just a quick overview of what I'm going to be talking about. So I'll give you some background. We're based down here in the Eastern Cape near Gqeberha. We're based at Grahamstown, actually, or Makanda at Rhodes. But most of our study sites and also the marine invertebrates we collect are around the Algoa Bay area. And the second step, I'll just describe how the Algoa Bay DOM experiment came to be and then discuss the results. All right. So to our background, I mentioned we do a lot of research on marine invertebrates. So a lot of the stuff's actually through SciAp collected with an ROV. It's like a little submarine on an umbilical cord you push off the boat. It's got a little collection arm. And then basically someone can operate with a joystick from the boat, can operate the ROV to then dive down and collect sponges. We've got a collection of several hundred sponges, many of them extracted already and accessioned into an extract library that we're assembling. And then we assay these extracts in the first step. It's just a simple assay of E. coli, Estuaries, and HeLa. just to assess possible antimicrobial activity. And now we've gone into fractionating a lot of these extracts to assay them again. But this is basically just to the background how we started using tandem mass spectrometry. And then basically what we do is if we have active extracts, we'd see which features that we get through molecular networking from the tandem mass spectrometry correspond to which compounds, which sponges have the same metabolomic profiles, which ones have different ones, and use that to inform our steps in terms of purification, because that's our final goal. We also do on-site here, compound purification. And then after the marine invertebrates, we branched into marine microbes from sponges, from seagrass, from soil, from water, and also some terrestrial microbes near water bodies. And then again, we use the molecular networking through GNPS based on tandem mass spectrometry to sort of dereplicate what type of compounds do we have present. And I just put some examples of compounds we quite often encounter here. All right. Yeah, just on the background, I'm not sure how familiar everyone is with mass spectrometry, or tandem mass spectrometry in particular. So I just made a quick slide of the process, how to acquire tandem mass spectrometry data. It's basically just injecting a sample onto an LC-MS. Chromatography, I think everyone's familiar with that. So basically our samples get separated. They enter our ion source on the mass spectrometer. We get a bunch of ions. They fly through the mass spectrometer and we record our mass spectrum. And then each ion basically will provide a distinct mass signal. And then in MS2 mode, basically, so tandem mass spectrometry, it's made up of normal mass spectrometry at MS1 level, which we call a survey scan. And then of each of these ions, we want to fragment them to get something like a fingerprint. And how that works is shown on this slide, for example. So we have in our ion source, continuously we create still the same ions. And then in the mass spectrometer, they're filtered to only let one type of ion through. Those ions get broken up, make fragments, and we get a fragmentation spectrum. And this is basically characteristic for each compound to a certain degree, and it works as a fingerprint that we can then use for database search to compare to spectra of known compounds, or to basically match them to known compounds or compare them to known compounds that are similar in structure, and thus we get some insight on potential structures for ions we observe in our experiments. All right, our data, essentially, I use MZMine3 to process the data. What it's basically of at the simplest level is just a sequence of MS1 scans or survey scans, and then fragment scans for particular so-called precursors selected for ions that are selected for fragmentation. Data is really a sequence of mass spectra, being one MS2 spectrum, and a bunch of spectra, and then again an MS1 spectrum, and then a bunch of 4. So essentially, then through MZMine we process this. We get 2 types of files. The feature quantification table. So this is essentially just like an Excel sheet, a spreadsheet or data frame, just shown on the left here. And each feature that we detect has a number. It has a mass, obviously, or mass to charge ratio, the retention time, and then the abundance of that feature in each sample that we're looking at. And then at the same time, through processing data in MZMine3, what we get is a second file, which is a spectra for each risk here, basically. And these are the fingerprints that we can then use for database search. Then here I just have a quick example for those who are not, or overview for those who are not familiar with GNPS. So what we do then is we have, this bunch of MS2 spectra for each feature, and we search them through GNPS. We upload them to the GNPS and get a certain similarity score. For each, basically pairwise, each spectrum, MS2 spectrum in our data set is compared, and from that, a molecular network is assembled where each node will correspond to an MS2 spectrum or an ion, and then these nodes are connected by edges based on how similar are the MS2 spectra. So you'll set like a minimum value, it's called a cosine value, and usually you can set between 0.6 or 0.7, 1 being identity, and then towards 0 being mostly any.
I think we're losing you, Dr. Kalinski. Oh.
I think he's kicked out. Oh, there you go.
When was I cut off?
I think just turn off the camera. I think that'll help a lot.
Let's see.
Yeah, so next slide.
You did hear me on the GNPS slide here, yes?
Yes, that's where we lost you, midway there.
Okay. All right, should I go over this again? I don't know, because I'm not aware when it cut exactly.
I think, yeah, that's an important part. Let's just go over that again.
Yeah, so, okay, let me pick up here again then. So we got MS2 spectra for all features that we detect on our samples, which should correspond to a particular ion ideally. And for, our MS2 spectra are like fingerprints, basically. They're characteristic of the structure of each ion. And then what we do with GNPS, basically an online platform we can use to analyze sets of MS2, MS, or fragmentation spectra. Those are all synonymous, by the way. And what we do is we upload our set of, and then for each pair of
Run again.
Dr. Zelinsky, your audio has disappeared. I think Makanda's having technical difficulties. We can still see your slides.
Can you hear me again now?
Ah, there we go. Your audio's back.
I don't know, it was on. I think that the AG-ROM isn't great right now. Sorry about that.
Okay, no problem.
Yeah, okay. So we basically GNPS creates a molecular network for us by comparing our MS2 spectra in our data set to each other, and then it compares also our MS2 spectra to spectral libraries to then annotate or putatively identify known compounds through spectral matching. Okay, and GNPS is basically just an online interface. You don't need to install any software, and this is what the start page looks like. And then, yeah, we employ this, as I explained before, to dereplicate, for example, antibiotics in microbial extracts. And then we use Cytoscape to visualize these networks, to color them in and everything. So you can download your files from GNPS and visualize in Cytoscape. All right. And now we can get to the actual part of the DOM analysis. So basically how this work started off was actually inspired by work done by Daniel Petras a couple of years ago in 2017 and 2018. He collected samples along the San Diego coast and into the San Diego River mouth and into the San Diego Marina, and then analyzed those using Tandem or LC-MS. And basically what he found, so he did this collection at two different time points, once before a rain event and once after a rain event. And what he found is that before the rain event and after the rain event, the LC-MS data was able to distinguish between the time points, which was not so surprising. But he also found that after the rain event, the sites that you see up here in the PCOA that correspond to the river mouth and the marina are clearly distinct from the rest of the sites. And he was able to relate this to xenobiotics that likely enter the river as runoff during the rain event, and thus cause this shift in the metabolites that you see in the after the rain event in the water samples. So, and then after Daniel finished this collection, he came to visit us in South Africa, actually, the year he finished his collection. And then he met with Dr. Xavier Sivenundo and Dr. Matcha, and they had an idea of doing a similar study in Algoa Bay. I was actually still a PhD student at that time, so I only got involved on the project a bit later. But yeah, what you see here basically is a map of Algoa Bay, and then we have our Sundays River and Swartkops River, and Swartkops River runs through Tabeja, and it's basically metropolis, so it runs right through the city and a lot of settlements, whereas Sundays River runs through farming areas, especially upstream. It runs through Edo, which is one of the biggest citrus farming areas in South Africa. and I think even worldwide. So there's a heavy agriculture going on there. All right, so here what you see is just as sampling sites. So we took 9 sampling sites along Swartkops River, 8 against Sundays, along Sundays River, and 8 across the bay. And then I've also put the dates of the sampling there, and you can see they were all sampled at different months. And it's a bit of a drawback of the study, but basically when the study set off, it hitchhiked on ongoing sampling operations. So we basically was set out to be a pilot study to just see what can we do with this technology, what can we see, how can it help us in the future, and then inform us for actual ongoing projects that we're doing now. All right, and how it worked is, so we went out, I was a little bit involved there, and we collected water samples in these plastic bottles. So all it really involves is putting a nitrile glove on and dipping them into the water. So we collected surface water. Then right after collection, they were brought back to the lab and they were subjected to solid phase extraction. So essentially we just apply a vacuum to this manifold here with our solid phase extraction cartridges up on top. And then we suck the water samples through the cartridges in the hopes that our organic compounds of interest stay adsorbed. And then we stored those at -80 degrees. And then they were actually shipped across to San Diego, where they were then reconstituted and analyzed on a Orbitrap. And that's basically what you see here, the exact instrument. And what's important is this data was analyzed using the exact same methodology as Daniel had used for his San Diego samples priorly. And then here on the left, you just see a set of tools that are used for the analysis of the data. Okay, so in the first step, data was pre-processed through MZMine3, and then I used QIIME2 to produce a principal coordinate analysis with a cosine metric, which you see on the right here. So we essentially got the data, analyzed it through that, and then to get a first overview of what's going on. And we could right away clearly see that the samples from the different sampling sites cluster together, apart from one sampling site down here, SW5, which is our river mouth in Swartkops River. So this site is more associated with the marine samples, or less dissimilar to the marine samples than to the urban samples. Urban, this would be Swartkops River, and yellow, this is our Sundays River. So it was the first observation that we could distinguish this. What I did in the next step then, so upload the data to GNPS, and then I got we have about 4,000 features altogether, and we got matches through GNPS for more than 200 of them. But now I had to manually curate them because sometimes you get matches that are basically false matches, so you can't trust it blindly. I manually curated the results, and I found about 60 features that were of high quality when I look at the spectrum matches of such quality that I'd be confident that I could convince peer reviewers for the study to be published that they're actually a decent match. All right, and then I basically used QIIME2 to draw up this the list of filtered features that I was able to annotate into a heat map. What you see on the left are the Sundays River sampling sites, in the middle Algoa Bay, on the right Swartkops River. And then what we found, I did a clustered heat map. Basically what we found is that all these pesticides up here clustered together, and they're predominantly present in Sundays River. So particularly of these imazapyr, pyrimethamine, and thiabendazole, there are pesticides used in citrus farming extensively, and particularly for citrus. And Sundays River catchment area is known for heavy citrus farming. So this made sense that we observe these here. And then basically the second block of labeled here, this was matched to mainly pharmaceuticals, and those were largely or at high levels present in Swartkops River, as opposed to Sundays River. You see, I put some little stars here as well. These are compounds that were matched to authentic standards that were used in Daniel Petra's study of San Diego samples. And we were able to actually even match the retention times and the MS2 spectra from our data, from Algoa Bay, to that in San Diego, since it was run on the same instrument, same methodology, everything. All right. And then, yeah, here, this is the other 30 features, basically, that we were able to annotate, which comprise of urban pollutants. They were also mainly present in Swartkops River. And then a second block of mostly natural products, and this is mainly beta-ene lipids, phosphocholine, and domoic acid, and a couple of other compounds that you can expect to see in marine water. So this is basically the part of the features that we're able to annotate using GNPS. All right, then I employed the molecular network to kind of look for molecules and their possible decomposition products. So I had one match here, for example, lopinavir. It's an antiretroviral drug. Then I looked at the molecular network. I did this for a couple of the annotated compounds to just see, can we find maybe new decomposition products? Then what you see on the right here is basically the spatial mapping data. So it's the LC-MS feature abundance per sampling site, and then mapped with colors onto the actual map of the sampling sites, with yellow and light colors corresponding to high levels and dark colors corresponding to low levels. So and what we found is, for example, feature B here, which shows quite a similar distribution to lopinavir. They actually produce the exact, like near identical fragmentation spectrum, same mass, but then when I looked at the XIC, so the extracted chromatogram, I could see that both of these features, they're actually distinct. It's a distinct peaks, so they're real chromatographic features we're observing. And then I theorized like what might be going on there. Then when I looked at the map here, I could see, okay, the one of the features occurs later downstream of the other one. So then I hypothesized might be a hydrate of lopinavir, basically extracts water when it enters the river. And then upon injection on the mass spectrometer, once it reaches the ASI source, might just eliminate the water right in the source and then convert in the source to create actual lopinavir, thus resulting in us having identical MS2 spectra for these two ion features here. And then the notion would also be supported by the difference in retention time, because our feature B here elutes slightly earlier. So if it had an extra abstractant and an extra water molecule, we'd expect the polarity to increase and it to elute somewhat earlier. And then we also had a look at some of the other related compounds. So I'm just looking for my mouse. There we go. So there's, for example, feature C here, which I was able to match according to the formula to known decomposition product of lopinavir, which is called oxolopinavir. And one thing we found when we looked at the spatial mapping is that for oxolopinavir, it seems that might be a little bit more persistent in the environment, since there's not as big a difference between levels in the bay and in the river for this decomposition product or putative decomposition product. And then we also had a look at some of the other features that were similar to lopinavir, and then we found this feature D here, which we hypothesized maybe a new decomposition product from lopinavir. Hasn't been described yet, and that notion may be supported by the fact that we see similar distribution distribution across the river for lopinavir or putative lopinavir and putative decomposition product. So we do that for a couple of pesticides and stuff we observe as well. I just showed you one example. Sorry. It's just not letting me switch the slide right now. Can you still hear me?
Yes, we can still hear you.
Great. I was worried. Maybe I was already going on for 10 minutes and I'd lost you again.
No.
All right. Okay. So then another observation I made, I looked at different ARVs that we were able to putatively identify. Now we have efavirenz, for example, nevirapine, emtricitabine. And I found that actually it seems the point sources for these different antiretrovirals seem to be at different points. I also included methamphetamine here as an authentic standard, which is at our highest site here, the point source. But you see for the antiretrovirals, they seem to have different point sources. And I wasn't quite sure how to interpret this. And actually these point sources, they also correspond to wastewater treatment plants along the river, like right at those three sites where you see down here, here, and here. And then after speaking to some people, I mean, we're all aware that South Africa suffers from HIV epidemic. And basically what I could gather so far is if you are prescribed antiretrovirals and you from a public clinic, then you will go pick up your antiretrovirals at your local public clinic. So what I suspect here may be that just different clinics use different regimens of antiretrovirals, and we see this in our DOM data. But I'm not entirely sure about this. If you have any idea or input, I will greatly appreciate to hear. after the talk. All right, and yeah, now to confidence levels and structure annotations. So in tandem mass spectrometry, basically there's different levels of the quality of an annotation. With our level 1 annotation or gold level basically being present when we have a compound and we match the retention time, the mass to charge ratio, and ideally the MS2 spectrum to an authentic standard. So that's what happened, for example, for methamphetamine, where it was run the authentic standard alongside the samples, and we got identical analytical data. So that's, then we can talk of identified basically. Where we have a couple of these, it's 5 or 6 altogether, as I've shown. Then level 2 annotations, that's basically what we get through GNPS, where we just match the mass to charge ratio and the MS2 spectrum. Now, that being said, if you look at the data I just presented you, we have level 2 annotations for a lot of the pesticides and the antiretrovirals, for example, but also we have the context of the antiretrovirals being present in the urban impacted river, or particularly present in the urban impacted river, whereas the pesticides are enriched in the agriculturally impacted river. So we sort of have a context factor that would lead me to argue that maybe we're at like a level 2+ annotation or something like that with them. And then a level 3 annotation, that's basically just if we're able to match our compound to compound class. Like we can define the ring fence what compound class does our ion belong to, but we don't know the actual structure. And then level 4 is if we just have a reproducible signal. It's a real signal, we're observing a real peak, but we don't have any idea of what compound class is it or what structure we're dealing with there. So that'd be level 4. Okay, and then we want to dig, basically we have a lot of level 4 data so far. Some we dereplicated to level 2 using GNPS, some we had the authentic standards. Now we want to get more of the level 4, not even annotations, but more of the level 4 signals into level 3 annotations. So how we do this is we use Series 5. So Series 5 is an in silico tool set consisting of 4 modules. And they're mainly concerned with predicting the molecular formula based on MS1 and MS2 spectra, and then predicting structures as well as predicting compound classes. So in the next step, what we did is we used Canopus to predict compound classes. So essentially all you do here, Canopus is even parameter free. You just upload your set of MS2 spectra and run it. Okay, and how that works is basically they trained a deep neural network using structural libraries and spectral libraries, and then of known compounds, so they knew which compound class they belong to, and then they use, now when we use Canopus, we use this deep neural network with our own MS2 spectra and predict the compound classes. So I did that. And first my reason for doing that was I wanted to see how does Swartkops River Site 5 here at the river mouth differ from the rest of the river and from the bay, to just get an idea of why it does cluster with the bay samples. And what you see at the top here is the average of all Swartkops River sites. The bottom here is Swartkops River 5, at the left Algoa Bay, and at the top here is Sundays River. So and one thing I saw is that it seems in Algoa Bay and Swartkops River Site 5, they're enriched in features and likely to be fatty acids. Whereas in the river, we have more polyketides that decrease to river Site 5. And you can see that Site 5 is somewhere intermediate maybe of Swartkops River and Algoa Bay. And then in Sundays River, we found mainly alkaloids to be, most of the signal abundance was related to features predicted to be alkaloids. And you have these 3 really abundant features here. And these are actually imazapyr, pyrimethamine, and thiobendazole. So the same compounds that we did actually get annotations through GNPS for. And then when I saw that the signal intensities were so high, it sort of sparked the idea in me that I'd at least want to get an idea, like what type of concentrations are we dealing with here? Like, to you know, get a ballpark estimate of really where are we at in terms of concentration levels. So I did some literature research, and I found this paper by Janos Ligand et al. From 2020, and it describes an app called Quantum. And basically how it works is if you have a bunch of authentic standards and a lot of features in your data set that you don't have any authentic standards for, you can estimate the concentration of the standardless features as long as you have a structure prediction. So the tool needs you to propose a SMILE structure, which we now for our data derive from GNPS. And then the tool basically, through machine learning, predicts what is the likely concentration within about a 6 times area. What is the likely concentration of the features in your data for which you don't have authentic standards, based on the response of the instrument towards those where you do have authentic standards. So I just made this little picture there for it. So it's basically, we have some anchor compounds, and using the model, we hit them in and estimate what concentration is present for our compounds where we don't have authentic standards. So we did that. You see here at the top the concentrations for our authentic standards, and then the predicted concentration for our annotated features. And then I looked at the predicted no effect concentrations for most of them. And just to see, so the predicted no effect concentration, it's a concentration below which you don't expect a compound to have a biological effect in an environment according to model organisms it was tested against. So for aquatic systems, that's Daphnia, some algae, and model fish. And then if it's below that concentration, you wouldn't expect it to have a negative effect towards those test organisms. All right, and then it's basically just like a rough estimate of what concentrations will be harmful to the environment and which won't be. Then I compared those to the predicted concentrations of our annotated features, and I found for 3, thiabendazole, imazapyr, and lopinavir, that considering even the error of 6 times quantification, that we are possibly in a range where concentrations exceed predicted no effect concentration. And particularly for imazapyr, if you see here, our predicted concentration is 300 nanogram per liter. So if we say 6-fold error, maybe it's only 50 nanogram per liter, but the predicted no effect concentration is 15 nanogram per liter. So this would basically earmark for us now that we should check in the river, like what's the real concentration of imazapyr, because we have some indication that it might exceed safe levels. All right, and then we went a bit further with the data. So this is also still ongoing progress. I'm not exactly sure what in the publication that I'm busy writing, what I'm going to include in that. But basically what we did here is just a 2-sided t-test plotted as volcano plots then of the rivers versus the bay. Importantly here for rivers, the river mouth site 5 was excluded. Yeah, we compared rivers versus bay, and then what we see here is I've just shown the most significant feature. And then what I'm busy doing is I'm going through the different features and then using CSI Finger ID, which is another tool from Series 5, or another plugin which allows structure prediction to predict what type of structures am I dealing here with the significant features. And then, for example, we have one terpenoid here. It's most significant feature, and it's present mainly in the bay. And then, yeah, if you see here in blue, that's basically the part of the structure series and CSI Finger ID are confident they can predict. So we are suspecting we're dealing with a similar structure to this one here. And then we're basically going through all of the most significant features to see where do we get good predictions, and can we make any statement in our manuscript regarding that. Same thing we do is in the comparison of Swartkops River versus Sundays River, where our most significant feature here is some type of purine. And then you see it's present in Sundays River, not present at the other sites. So basically working through that to better characterize what are the differences between the different sampling sites. Okay, and then as a summary, so using tandem mass spectrometry and LC-ESI-MS-MS, we were able to show that Swartkops River, Sundays River, and Algoa Bay exhibit distinct DOM compositions. We're able to putatively identify various pesticides in Sundays River and pharmaceuticals and urban pollutants in Swartkops River. Some of them extend into Algoa Bay, and our putative identifications are sort of strengthened by the context of pesticides being present in at sites where you would expect them, and pharmaceuticals vice versa. And then we were also able to identify potential degradation product of lopinavir through molecular networking. And lastly, we were able to predict concentrations for putatively identified features. And this indicates potentially concerning levels of some xenobiotics in the rivers. And then, yeah, lastly, actually, for the data that's collected, so on both rivers, previous studies have been done. like even mass spectrometry studies, but they were always targeted studies. So where someone would go with a set of known antibiotics and just quantify those. And now using this data for any future endeavors, we basically now have a repository of what compounds can we detect using this approach. And now if we want to go quantify, we can basically know already which authentic standards do we need to buy that might give us interesting results or that are worth studying. All right, and yeah, now we're basically expanding this project into, for example, mapping algal blooms in one of the rivers. And then we're also aiming to do some more future work doing coastal marine DOM analysis using the same approaches. So we have a few collaborations where we're looking in the future to do more of this type of work. All right, and that was it from me. Thank you.
Thank you very much for a very interesting presentation. So, in every journal club, I ask our presenter to present their email, and you can see there on the last slide there that the presenter has his email there. If there are any questions for the presenter, you can either raise your hand and ask him verbally on the platform, or you can write it in the chat box. So please, we encourage our audience to ask any questions. So while we're waiting for questions, I have a question for you, sir. So you found a lot of ARTs from the rural area. Do you think there's direct dumping of ARTs, or is that from sewage runoff?
I think for the most part, it's actually it goes through the wastewater treatment plant for the most part. I mean, you will have runoff as well for sure, but I mean, most of that will be excreted in urine and fecal matter, so that lands in the toilet in most parts, then goes through the wastewater treatment plant. But what I've also been able to gather from people I've discussed this with is that the wastewater treatment plants, especially for antiretrovirals, it's known that they're not very efficient at retrieving them or filtering them out of the water, and they don't get metabolized much in the body. So basically, they come out pretty much the same way as they go in to a large extent. Yeah, and that's I think where they come from. We see some levels also in Sundays River, the agriculturally impacted river, but also important there is that in the farming areas there are also people living there, like a lot of farm workers and such. So one could expect also to get some input of antiretrovirals in that river. And then what I found interesting is we can even detect them in the bay. So that's like I've checked the data. If I just look at the bay water samples separately, like they actually produce the actual MS2 spectra. So it's not like a gap filling or imputation reason why we see them there. They are actually present in the bay as well, which I found surprising to like considering dilution and such things that we're still able to detect them there.
Yeah, it's a bit concerning to see that the one ART is 20 times higher than the lowest effect value. But yeah, so it's very high values of ARTs.
Yeah, so here, I mean, you also have to consider we have the error. It could be 6 times higher or 6 times lower according to the model. So how accurate that really is in the end, I'm not entirely sure, but it's just like a best estimate essentially. But also for this data, the way I estimated these concentrations. So because we have the SPE happening, right? So on the SPE, you don't always have 100% recovery. So if you load a compound onto the SPE and you elute it, you might end up with less than what you had before. So for all these predicted concentrations, I assumed 100% recovery rate. So it's rather like within the uncertainty, it's a rather low estimate. So yeah, it could be way higher or it could still be borderline okay, but yeah.
Another follow-up for your study would be to see what is the effect of ARTs on animal life.
Yeah, I mean we're looking to maybe do something like that in terms of collaborations. I mean it'd be interesting to tackle it basically from 2 angles, from a biological angle, like observing certain model organisms from the river and then trying to associate that to the drug levels. Something similar we're trying to do now in Sundays River. We're trying to look at the algal blooms and now also maybe how that's related to the presence of pesticides. So if we see the timing of the algal blooms somehow relates to the presence of pesticides, that'd be interesting. If either the algal bloom feeds on the pesticides or, yeah, well, I mean, we have no idea yet what's going on there, but that's sort of the project that we've started off now, the next follow-up to this.
Yeah, like you said in the beginning, it was an untargeted study you started off with, and you've created lots of hypotheses from your data, which is great. So you have a lot of potential directions to go.
Also, one thing that I actually did not mention is, I said here, we use structured predictions, right? So through series 5. So for me, before I started with this project, I did like use series 5 and CSI finger ID, but I never really had confidence in do these structures make any sense, right? So what I need to say to this is for the 60 selected MS2 matches, where I'm confident in that they're likely right, or have a good possibility of being right, for the 60 of them, 40 of them, the structure was correctly predicted through CSI finger ID. So independently of any spectrometry. In silico predicted 40 out of 60 were correct. And then those that were incorrect, it was because Sirius had actually not been able to annotate the right molecular formula. That being said, if you are able, my notion from that is if you're able to annotate the molecular formula correctly, then CSI finger ID can produce really good results in terms of structure prediction. So basically what that means to me in the future, like, or moving forward, as instruments get higher mass accuracy and higher isotope pattern fidelity, the utility of especially the in silico prediction tools is going to be a lot bigger.
So you use lots of tools as well. So anyone who's interested in the tools that our presenter showed and wants to find out more, please contact him. I'm sure he'll be able to share with you. There's a question in the chat from Dr. Kwaji. So he says, thank you for such an awesome presentation. I want to ask, are you also considering the possible secretion of bioactive compounds? So antimicrobials by marine invertebrates and the possible effects of those deposited pharmaceuticals on the marine ecosystem?
We're not at this point, really. I mean, we do, in the bay, we do marine invertebrate sampling, but that'd be an interesting way to look at things. But I guess we'd need like a very focused sampling strategy to engage with that. But what I can say about this data set, I actually detected, I can't remember the name, but I detected a sponge natural product as well in the bay, so which I also found surprising to find that from simple LC-MS analysis of the DOM. So I think maybe some of the marine invertebrates excrete the secondary metabolites into the environment, and thus we see them. But I'm also, it's a putative annotation that I had for that compound, so it's not, also wasn't included in the presentation here. But yeah, so it could also be a wrong annotation, not verified. But I did see some features that were matched to marine natural products from marine sponges specifically. Yeah. But I mean, with this approach, you could do a lot of stuff. And I think also, I'm even thinking maybe there's people who are sitting with data, you do a targeted experiment. If you do LC-MS, maybe you've still done a data-dependent acquisition method, and you have data that you can analyze through these tools, basically reanalyze or answer additional questions to what you did already in a targeted study.
Okay, are there any other questions? There's another one. Awesome. So Sam says, thank you for the presentation. You highlighted the influence of the wastewater treatment plants on the ARV's inputs. Did you also consider this for other chemicals?
Not specifically, but this is actually a really nice suggestion. That's actually a very nice idea to look at the data in such a way, see which ones correlate with the output from the wastewater treatment plants. Yeah. No, thank you for that, but we haven't considered it in depth, but I will now.
Another comment, interesting stuff. Can you explain a bit more about the quantum analyticals tools? It looks very interesting.
Yeah, I'm not too well versed on how exactly it works at this point, but you basically uses a machine learning model and then uses tables of ionization efficiency for different compounds, and then what you do is you to quantum. So quantum is actually kind of half paid. You have to. It's not completely free. Let's put it this way. You can, I think, quantify 50 features a month. I mean, you can contact the developer as well, I guess, to ask for more if it's in an academic context. But what you do is you just have your sample. You enter in a spreadsheet the concentration of known compounds you have with authentic standards and their SMILE structure, and then the same for other features you have. So I'd have other feature, the signal intensity. So there you just add the signal intensity, not the concentration, and then the SMILE structure, which are derived from the GNPS match, and then you just run it through the tool. It's basically trained a model, a machine learning model on ionization efficiency data for various, like thousands, 100,000. I don't know exactly how many compounds, but many.
So if you can go to your last slide, please, just so everyone can see your email.
Okay, this one now.
Because we are going to upload this onto our YouTube channel, so anyone can rewatch it again if you see an interesting tool there, or you find information from this presentation relating to your work or anything like that, or you want to collaborate or ask more questions, please contact our presenter. Yeah, we encourage communication with the presenters and post-presentation conversations, discussions.
Yeah, I'm also looking to potentially give an online webinar on feature annotation next month, maybe. So if you have feedback of what you're interested in also in terms of that, or what basically for me to kind of get an idea what are the needs and wants of the community, then you're very welcome to get in touch with me about that as well.
I think we'll definitely take you up on that offer.
Definitely.
Yes, you presented lots of interesting tools there for our LC-MS crew. I know Fidel will be interested in a few of them as well.
Yeah, I think Fidel, you already used a lot of them. I saw some of your publications where you employ them.
There's a comment here, so any links to the YouTube channel. So you can just go to YouTube and type Metabolomics South Africa, and you'll find us there. And after each presentation, the communications committee, they upload the presentation there. So we don't have a link yet for this one, but they will be uploaded shortly, and we'll include the link for the next presentation. So you'll see the communications, each email that they introduce the new talk and they give a link to the previous talk there. Abby Stoller has a question. So she says, thank you for your presentation. I was just wondering if you considered how the aquatic organisms and microorganisms adapt to this pollution.
Yes, so initially the sampling we did was actually accompanied by microbial analysis as well, microbial community analysis. And I've had a look at the data, but there's a lot of interplaying factors coming from the tide and inflow from different points and things like that. That's difficult to draw any correlations at this point. And I think that's something we're going to tackle in future studies, because you want sort of, I mean, to assess something, you ideally want something to compare it to. So, yeah, it's like we have the data. I'm still looking into. We're probably going to work that into a second manuscript, I think, because it's a if we even able to find any meaningful information out of that. But basically, at initial sight of the data, there are a lot of factors that influence the composition of the microorganisms. Same as to the presence of the metabolites. So we haven't been able to, at this point, draw any clear picture from that. But there is microbial data actually on the full set.
Okay. Are there any last questions for our presenter? Dr. Kalinski, thank you so much for such an interesting presentation, something we're not familiar with, but definitely the analytical platform we're familiar with, but the output, the way he presented it was very interesting. The number of tools you use was very interesting as well. So I hope you get a lot of additional output from this so you can publish on it and go forward with future targeted analyses on your project. So I think you have quite a lot ahead of you.
Yeah. Awesome. Yeah, thank you so much for the opportunity to present. It's nice to get some of our work out there.
Yes. And if you have any future results coming soon, we're more than welcome to present them on our journal club.
Thank you. Yeah.